The overall goal of this procedure is to achieve a standardized follow-up in the in vivo behavior of stem cells transplanted into the mouse brain. This is accomplished by first labeling the bone marrow-derived stromal cells, which co-express EGFP and luciferase, with fluorescent micron-sized iron oxide particles. The second step is to appropriately inject the MPIO-labeled BMSCs into the mouse brain tissue. Next, the survival and localization of cell grafts are followed non-invasively using bioluminescence imaging and magnetic resonance imaging. The final step is to validate the in vivo imaging results using post-mortem histological analysis.
This method can provide insight into the behavior of stem cells after transplantation into the mouse brain, but it can also be applied to other systems such as model organisms and studies of a disease.
Visual demonstration of this method is critical, as the animal handling steps are difficult to learn because intravenous and intracerebral injections are difficult to perform and contain certain risks for animal survival.
To begin this procedure, add 75 × 10 to the 6th glacial blue micron-sized iron oxide particles to a subconfluent growing cell culture. Incubate them for another 16 hours to allow particle uptake via endocytosis. Then, wash away the remaining particles. Allow the cells to grow for an additional 24 hours to obtain cell confluency and homogeneous distribution of the GBM.